These are predominantly held in place by non-covalent forces. One of the things that's really important is that you understand that membranes are not just barriers or walls. They are not static structures. They are very dynamic. Everything that I'm going to tell you about in this, we're listing all the structures of them, is spinning around in 360 degrees like a top. It's bouncing up and down, and it's moving literally in circles, in spherical motion across the cell. We call that a fluid mosaic model. The two primary, or really just the main constructs of a lipid consists of a membrane, consists of the lipids themselves, and then we have the proteins that are associated with them. We may have sugars involved in this, but they'll usually be attached in the form of a glycolipid or in the form of a glycoprotein. The two primary lipid components of the membrane, we have the phospholipids. They're going to be forming our phospholipid bilayer. And the other part that we have for increasing our membrane stability would be the cholesterol. So as a phospholipids, these are going to associate to form a bilayer. That's kind of a given, right? Because the bilayer is going to function to do two things. It's going to have permeability to it. Permeable, it's not going to allow things in or out. It does exhibit some selective permeability, of course. Certain things can come in and out and diffusing in small amounts, but for the most part, it's really just the larger macromolecules that we're going to have, keeping the polar stuff in and out, and then also nonpolar stuff embedded in the membrane. But it also is going to form spontaneously in a hydrophilic environment. So if we take it in water, they're going to form little things known as liposomes, or I like to just envision them as vesicles. If they are just not phospholipids, but just regular lipids themselves, they'd form micelles. Now, I've already kind of talked about the adaptations that we can have to the hot or to the cold, but just for review, remember that we can have things to deal with varying degrees of unsaturation versus saturation, and we can have changes in the carbon atom length. Cholesterol does two things, though. Cholesterol contributes to the overall stability of the membrane, maintaining it at a specific level of fluidity. This works both really good in hot environments and really good in cold environments. What we haven't really mentioned much of, and this book doesn't really mention much of, is the formation of lipid rafts. For the stability part of it, we have the OH group, exhibiting this amphipathic nature of it, obviously. So it has the OH group there, and then the overall just the nonpolar hydrocarbon. This is going to be associated with the polar head groups, whereas this is going to be associated with the nonpolar tails. Now, lipid rafts also contribute to the formation of stability. They can make things very rigid because you have cholesterol. Basically, that's not how you spell rigid, is it? I don't know. You have basically cholesterol coming in and interacting with those fatty acid tails, really kind of hindering the amount of movement that can take place. But what I think lipid rafts are really good for is they're useful mechanisms for signaling. They're very rigid structures. They're not going to be bouncing around there. So protein molecules that are involved in this can associate with lipid rafts. A guy by the name of, or a professor by the name of Kai Simmons, there's an iBiology podcast. You should check that out. The proteins, we have two definitions for them. There's the peripheral proteins and then the integral protein. The peripheral proteins, obviously, if we were to just kind of visualize this for a second, if here's our cell, here's our phospholipid bilayer membrane and all the other junk we got inside it, there's the nucleus, whoop-de-doo. If we were to look on the outside areas here, just on the surface here, we'd have peripheral in the periphery, right? So they're either on the outside, on the extracellular sites, or they're bound to the intracellular sites, but they don't have any specific, or they are, directly selected to one specific region. So whenever I say peripheral, I'm talking about the fact that they're going to be binding with the polar head groups of the phospholipid. In case you didn't know this is non-covalent, right? So this is hydrogen bonding. Now sometimes peripheral proteins, this is kind of weird, and this is kind of an exception to what I just talked about, though, that they can form covalent, can be covalently bound to the fatty acid tails, and they're acting as an anchor there to them. But the overall structure of the protein is remaining either on the outside or on the intracellular space. The structure of the protein does not completely dip into that hydrophobic region. So for an integral protein, however, these things are going to be anything that has basically associates with both. If it associates with both the polar and the nonpolar sides of the phospholipids, and the protein itself, the whole functional unit of it associates with it. We have anything that dips into the nonpolar fatty acid region from the outside or from the inside. And that is a long definition to write out, I know. So these integral proteins, they may or they may not be transmembrane. Okay, so if we were to just kind of playing along in this little analogy here, we have some phospholipids here, phospholipids there. So if we have a protein that's here, like this little green thing here, this would be a peripheral protein. I'm drawing in this direction. If we have a protein that is, and I'll do it in blue, that looks like this would be an integral protein. Or if we had a protein that was like this would also be an integral protein. This is also a transmembrane. I'm really dragging this down here, that both the peripheral and the integral proteins have really unique structures. I think you can imagine what type of structures these would be. If they're non-covalent, we're going to have both the alpha helices and then the beta sheets, or beta barrels. And the majority of the time we're going to be using an alpha helix as opposed to a beta pleated sheets. That's because alpha helices are a little bit more thermodynamically stable, because the structure of an alpha helix is really more three-dimensional than that of a beta sheet, which is kind of flat. And I'm using those terms subjectively, no, not literally, but I'll just kind of reiterate what I'm talking about here. So with the alpha helix structure, let's say that we have ourselves just a bunch of polar phospho head groups here, polar phospho head groups here. For a alpha helices, the way that we're going to have this arranged is kind of from the inside we're going to have these little helical-like structures here. I'm not going to really stress the right-handed or left-handed nature of them, but I'll just draw them like that. And in the middle, these green that I've labeled in green, and I kind of haven't drastically shown them at the scale though, these are going to be the nonpolar amino acids, because the hydrogen bonding that way is located strictly on the inside, shielded away from the polar fatty acids tails, whereas for the rest of it though, if I could switch to gold here, these regions here, these are going to be containing the polar amino acids, which we usually have as attachments there, or they can be looping out or whatever. That's not really important so much in determining our alpha helical structure. But if we also to visualize how this works, just kind of this part here, really, I guess, trying to drive that point home. So here we have, and I'm going to draw this blue areas here as the top part, if you could imagine, of the phosphate heads of your lipid membrane. So these guys are all like the polarized part. And on the inside of that, for the alpha helix, remember that's a helix, right? So it's going to have this kind of this coiling like structure here, like if you could imagine it being in there. On the inside of that is, and I'll do the hydrogen bonds in gold, is hydrogen bonding between those bad boys, right? So the hydrogen bonds are staying inside, stays in, away from the fatty acid tails of the nonpolar region. Hopefully that's doing some justice to it. I don't really feel like it is. Beta sheets are the exact same thing, but instead of they don't really exist in beta sheets. They exist in the form of barrels. And what I mean by that is, so say you have a porin molecule, which is what bacteria use to transport water in and out. So this is a porin molecule. This inside here has to be polar in order to allow water to pass through it. And the way that I'm going to do that, very similar to what I did with these alpha helices here, is I'm going to have a beta pleated sheet, I'm going to switch colors to green, forming a complete little protective beta barrel around it. Remember that they're kind of flat around these little sheets. Imagine taking a piece of paper and folding it up like that. And so there's this interlocking here, so that I have that structure concave there, to where I have the nonpolar affiliation on the outside and then the polar type affiliations on the inside.